Today we'll finish with cloning, and we left it here that whenever we have a vector with an insert of interest, you'll see this IOI throughout slides. That means insert of interest. And we mix it with bacteria. Some of those vectors with our insert of interest will make it into the bacteria. But obviously, not all of them will pick it up. So the way we select for those bacteria that did pick up the plasmid with our DNA of interest is using antibiotics. If we subject bacteria without any plasmids in them to antibiotics, they will all die, right? So that's what's shown here in that plate where nothing grows. If you don't add the antibiotic to the culture plate, obviously every bacterium will replicate and it will form, the whole culture will form what we call a bacterial lawn, right? On the other hand, if some of the bacteria picked up the vector and the vector carries an antibiotic resistance gene, some of those bacteria will be resistant to the antibiotic and they will be able to proliferate and grow and make a colony on those plates with the antibiotic. And in those plates, we say that only transformants grow. Okay. And this is a cartoon. I don't know if many of you had an opportunity to see what this looks like in real life, but here in the opening slide, I have a picture of what a plate with colonies growing in them looks like, right? So it's those creamy dots. Sometimes they come in different colors and shapes, but for the most part, there'll be these dots on a plate. So the question now becomes, I mentioned that we introduce this insert of interest into a vector, but the question becomes, how do we do that, right? And that's what we'll discuss next. From a sort of a bird's eye view perspective, a very general perspective, the general strategy is you have your insert of interest somewhere in a very long DNA molecule, typically a natural chromosome in a biological species. And then you have your vector, and the vector will have that antibiotic resistance gene in it. So the general strategy is we use enzymes to cut the insert of interest out of its original source. We use enzymes to cut the circular DNA vectors open, and then we introduce that insert of interest into the vector, and we stick everything together, and now we have the vector plus insert that we can then offer to bacteria for some of them to pick it up and start making many copies of it, okay? And one thing that we mentioned on Tuesday is that these vectors also have an origin of replication so that the bacteria can make copies of them as they divide. So it's the general strategy. Now let's go through some of the details. To do this cutting that I mentioned before, the cutting out of the insert from its original source and the cutting open the vector, we use a very special family of enzymes, typically a very special family of enzymes that we refer to as restriction enzymes. Sometimes you'll see them as restriction endonucleases because that's what biochemically they are. They cut nucleic acids somewhere inside of the molecule. But for short, we call them restriction enzymes. And what these restriction enzymes do is they recognize specific sequences of nucleotides in DNA, and then they will go and introduce two enzymatic cuts on both strands of DNA, effectively separating that DNA molecule into two, right? So let's say this enzyme here, I'm going to zoom in, the enzyme here recognizes this particular sequence, AAGCTT. Biochemically, it recognizes it using some of those same tricks that we mentioned when we talked about transcription factors binding to DNA. Some parts in the protein structure of these enzymes can interact biochemically with this particular sequence, AAGCTT. And then there is another catalytic site in these enzymes that will go in and introduce those two cuts. And the way these cuts work is by breaking the phosphodiester bond between these two adjacent nucleotides. Now, most enzymes that we use in labs, most restriction enzymes that we use in labs for cloning will introduce these asymmetric cuts such that it's not the exact same pair of letters facing one another that gets cleaved or that whose phosphodiester bonds are cleaved. But instead, there is these asymmetric cuts such that there is one cut on one side and then on the other, generating these what we call protruding ends. And why these matter, we'll discuss in a second. But for now, let's just imagine these enzymes that have the ability to recognize certain words in DNA. And when they see those words in DNA, they will go and introduce these two cuts on both strands of that region of the DNA molecule, such that it's asymmetric and it leaves these overhangs or protruding ends. One characteristic of restriction enzymes is that they normally target palindromic sequences. Palindromic sequences are those that read the same way, one direction or the other. So some fun palindromic English words, radar, madam, level, civic, kayak, any of these words, you can read them backwards and they read the same way. So that's what a palindrome means, and we borrow that term to describe what these restriction enzymes do in cells, and that is recognize sequences that read the same way, one way and the other. And to demonstrate that, we can use the same example. So I told you that this enzyme is recognizing AAGCTT. If I were to read this region of DNA backwards, it would read AAGCTT. So it reads the same way in both directions. And the thinking is that the reason for that is evolutionary, or it's explained by evolutionary biology. Restriction enzymes are defense mechanisms for bacteria. They use these enzymes to attack any DNA that is not their own and cut it into little pieces. And it is easier for a restriction enzyme to recognize its target sequence if it's a palindromic sequence, because now the enzyme doesn't need to approach the DNA molecule in any given direction. It can approach it from either side and it will recognize it and attack it. Right? So the thinking is that there was some evolutionary pressure for selecting for these enzymes that can recognize palindromic sequence, because it didn't matter which direction the enzyme encounters the foreign DNA, it would digest it anyway. Down here, I have a slightly different depiction of what this looks like. So again, we're focusing on this sequence here, AAGCTT. And what this depiction down here emphasizes is the fact that the enzymes, what they do is they break the phosphodiester bonds between adjacent nucleotides, such that now all there is left are these very weak hydrogen bonds between the protruding ends. So even though these molecules don't necessarily have to separate immediately after these two phosphodiester bonds have been broken, chances are that in a biological context and at regular, like room temperature, they will, because these 4 hydrogen bonds, or these 4 base pairs between the protruding ends, are not enough to maintain these two molecules together. For all intents and purposes, if this happens, let's say, inside of a bacterium that lives in our gut at 37 degrees Celsius, if the restriction enzyme cuts in a sequence like this, AAGCTT, the 2 fragments of DNA will separate. So far, so good? So that's what restriction enzymes do. They're like molecular scissors that recognize DNA at very specific locations, and they introduce these cuts such that they leave these protruding ends that we call sticky ends. Not all of them do that. There are some enzymes that generate blunt ends. Those are very, very useful for bacteria. They're a lot less useful for us as human beings in the lab, because by not generating those protruding ends, they don't leave us with this really cool feature of the overhangs, the sticky overhangs that I'll describe next. So these enzymes that generate blunt ends exist. They're sometimes used in genetic engineering and molecular biology labs, but they're a lot less common. Okay? The principles of recognizing palindromic sequences and introducing cuts in both strands of DNA still apply, and these enzymes do exist in bacteria for the same defensive mechanisms that we discussed for the other ones. All right, so how does this work? I'm going to walk you through the same cartoon that we had before, but this time I'm going to use these pseudo magnifying glasses to see what's going on at each step of the process. I mentioned that first we would use restriction enzymes to cut the insert of interest out. So what we can imagine is that we're using a restriction enzyme that recognizes target sequences, its target sequences, on both ends of our IOI. So what we need to imagine is that this insert of interest is flanked by two of these sequences that a given restriction enzyme would recognize. And if we take a look much closer, what we see is that at both locations, what happens is the restriction enzyme goes to work, and it generates these breaks that are asymmetric such that there is these protruding overhangs. I mentioned that you would do the same to the vector, right? So here we are cutting the vector. For this example, for this particular case, we're using the same restriction enzyme. So we do the same thing to the vector, and once again, in the vector, the restriction enzyme X, whichever that is, will recognize its target sequence, cut, leaving the overhangs.
For the vector, would it have two restriction enzyme like DNA parts, or would it only have one?
So for now, we're using one. We will, a little later today, we'll discuss how we can use two, and that can be advantageous, but for now, we'll leave it at one.
And then how does it determine like how long it should cut from the vector? Like, is it just based on?
It will cut at only that one position because the restriction enzyme recognizes that particular target sequence, in this example, AAGCTT. It sits there, cuts there, and leaves, right? So it's not like it's cutting and then digesting or chewing nucleotides off or anything like that. It just introduces that single truncation in the DNA molecule. So now we can imagine that we have separated the insert from the original source DNA, and we've cut the vector open. And in both cases, what we'll see is that now the ends of our fragment of interest will have these protruding overhangs, and same for both ends of the open vector, they will have these protruding overhangs. Now, what's interesting about this is that if we use the same enzyme to cut out the insert of interest from its source and to cut open the vector, when you pay attention, you'll notice that these protruding overhangs are complementary to each other, right? So we'll do it together. The protruding overhang for one side of the vector is TCGA, right? And then the protruding overhang for one end of the insert is AGCT. So A here in the insert will pair with T, G will pair with C, C will pair with G, and T will pair with A. And if you do the same on the other side, you'll recognize that the same happens on the other side as well. So we have one end of the insert being able to base pair with one open end of the vector, and the other end of the insert being able to base pair with the other end of the vector. And that's exactly what happens. If we mix these two together in a tube at a low temperature to allow for those very few base pairs to form, so this is typically done at around 12, 14, 16 degrees Celsius, so it's a cool water bath, these base pairs can form and be stable, such that now we've reconstructed what was or used to be the target site for these restriction enzymes, except that now these are hybrid molecules in that each half of that restriction enzyme site or target site is made up of part vector, part insert. And the same happens on both sides of this, right? Are you guys following this? Yeah? Any questions so far? No? Cool. So for now, very simple. So after there's been this base pairing of the insert of interest into the vector, what we would see if we were to look a little closer is that there is all of these base pairs between the corresponding complementary nucleotides, both molecules. But while there is let me change the color here, while there is all of those phosphodiester bonds linking the nucleotides in the insert together and linking the nucleotides in the vector together, there is still these missing phosphodiester bonds at both ends of this ligation site, right? Those phosphodiester bonds are not formed magically. right? They don't form spontaneously. So all that is holding the insert and both ends of the open vector together for now is very weak base pairing. And the only reason why the insert will stay there and not float away is because we're doing this at very low temperatures. But we can use this vector in living organisms at very low temperatures. At some point, we may want to put this into a biological system that requires much higher temperatures. And if we did that, the insert, these base pairs would weaken to the point that the insert would float away. So what we need to do is reconstruct those phosphodiester bonds that are missing between those neighbor nucleotides. And we've seen the enzyme that does this under natural conditions in living systems is an enzyme that we call ligase. It's the enzyme that will go and introduce those missing phosphodiester bonds between neighbor nucleotides when DNA is replicating, and it is being replicated in a fragmented manner through Okazaki fragments. We discussed that there is that interplay between DNA polymerase 3 extending from an RNA primer, then DNA polymerase 1 exchanging those RNA nucleotides for DNA nucleotides. But when DNA polymerase 1 leaves, it cannot finish the job and link the last two nucleotides together. So that's the job of ligase, which comes in and fills in those missing phosphodiester bonds. So when that happens, when those phosphodiester bonds are created between the adjacent nucleotides in the insert and in the vector, now the insert is covalently bound to the vector. And it won't go anywhere. It became part of that molecule, that circular DNA molecule, and it'll stay there. Okay? Became part of it. We're good? Yeah? All right. So, in practice, what happens is these vectors that we normally use for cloning inserts of interest in labs will contain what are called multiple cloning sites. In jargon, sometimes we call them polylinkers, right? And MCS for sure, multiple cloning sites. And what it is just a collection that was introduced there artificially by design, a collection of target sites for different restriction enzymes, such that as a user, you can take a vector and decide which restriction enzyme you'd like to use to open the vector up. And that decision obviously will be dictated by what restriction enzymes you might be able to use to cut your insert of interest out of its original source. So if the enzyme that is handy or convenient for you to take your IOI from its source is, I don't know, Hindi3, then you will go and cut the vector with Hindi3. If it turns out that you're much better off using eCore1, then you open the vector with eCore1. Right? So we can't really do much, and we'll revisit this in a second, but we can't really do much about what enzymes we can use to get inserts of interest out of their natural sources, because that has been established by evolution, right? We can't really decide what restriction enzyme sites should flank an insert of interest in its original source. All we can do is look, find one that we might be able to use, and then we make it easier on ourselves by creating vectors that have a collection of restriction enzyme target sites, hoping that one of the ones that we found in our natural source would be present in that polylinker. So that's just a cartoon showing in cartoonish fashion what a vector should look like. This is a real map of a real vector that someone might use in a lab. And as you can see, the MCS here has all of these target sites for all of those restriction enzymes. Basically giving you a lot of options to cut the vector open and hopefully be able to incorporate your insert. All right. So back to this idea that vectors will have a polylinker, an MCS, with several sites to choose from, but the insert of interest in its original source won't, right? So one of the things that we can ask ourselves is, well, let's say that I have this vector with all of those sites, and I'd like to use Hindi3 because it's a very reliable enzyme that has this very potent, very very faithful. It will operate under different experimental conditions. So I like to use handy 3, right? But what are the chances that I will find the handy 3 target sequence, AAGCTT, surrounding my insert of interest, right? I may find some of those sites around my insert of interest, or I may not. Now, the good news is that nowadays, for most species of interest in experimental biology labs, we have their genomes, so we can simply ask the question, are there handy 3 sites nearby surrounding my insert of interest so that I can cut it out and clone it in a vector? But in the old days, you could still try and calculate what might be the probability that this particular sequence will happen in a DNA molecule. So the way that we would have calculated that is simply asking the question, what is the probability that we have this particular collection of 6 nucleotides in that particular sequence? So the probability of that first being an A, what is it? 1 in 4, correct. So the probability of the first nucleotide being an A is 1 in 4. What is the probability of the second nucleotide being an A? 1 in 4. And you can see where we're going with this. So the probability of finding any particular sequence that is specific of a given length n, in this case 6 nucleotides, would be 1 in 4, or 1 over 4 to the n. In this particular example, it would be 1 in 4 to the power 6. When you run the math, you will find that, yes, it is very, very likely that in a complex genome, you will find many random sequences that will be like the target for handy 3. So many occurrences of AAGCTT. But they may not be exactly flanking your insert of interest. They may be much further away from its ends, in which case, if you wanted to use that enzyme, it's hard, because then you would be taking out from your DNA source a much larger piece than what you really need. So let's suppose that we're trying to use handy 3 and we find that there is a target site for handy 3 here and 1 here. Technically speaking, we could still use it, but then the insert that we would be trying to introduce into the vector is way more than what we really need. So what are the chances that your insert of interest will be precisely flanked by a given restriction enzyme target sequence? They're low. They're low. So that's where we bring in PCR again. And I'm going to walk you through this process by which we can use PCR to clone a portion of DNA into a different context. This was one of the uses that we listed on Tuesday, but we didn't go through. And essentially, the trick is you modify the primers that you use to amplify your insert of interest from its original source. So instead of cutting it out from its source, what you do is you PCR it out from the source using primers that have these little tails with the sequence that corresponds to your restriction enzyme of choice, right? It so happens that I changed the restriction enzyme here. That's my bad. So instead of being the AA-GCTT, it's a slightly different one. It's GAA-TTC. I think that's the one for EcoR1, I think. But let me walk you through the process. So you have your original DNA with the sequence that you'd like to clone into a vector, but there are no sites for a restriction enzyme that is convenient for you to cut the insert out of the DNA. So what you do is you do a PCR with these modified primers. Cycle 1, you denature that DNA source by heating it up. Then, part of cycle 1, the second step would be annealing, and you use these primers that have complementarity to both ends of the insert of interest that you would like to PCR out of this DNA source. And like I said, these two tails that are hanging out on the other end, on the 5' end of these primers, and you will see how they come handy in a second. The point is, by design, we're subjecting this DNA template to primers that do not have 100% complementarity to the sequence in the target. But I want to emphasize that that's okay as long as there's enough complementarity between the primer and the template DNA such that the free 3' end with its hydroxyl is still exposed on both ends. Because that is, and in case you're wondering, that's a 3' hydroxyl upside down. Because that is what Taq polymerase needs to start synthesis of DNA. The Taq polymerase doesn't care about what's going on in the 5' end. So if this 5' end is kind of flapping around, not bound to anything, Taq polymerase doesn't care. The only thing it cares is that there is a free 3' hydroxyl from which it can start adding new nucleotides following whatever the sequence of nucleotides is in the template. Okay? So annealing of the primers in the first cycle will look something like this. Then we will have extension. So during extension, there's going to be synthesis of new DNA in both directions. And because of what we discussed on last Thursday, during this first cycle of PCR, there's going to be one of these beyond pecans that goes way beyond the end of our insert of interest. But that's fine. This is just cycle 1. Then in cycle 2, we're not going to worry about what happened to the original sample DNA, the really long chromosome molecule. We know that those are going to continue generating beyond pecans with every cycle of PCR. But what we care about are these beyond pecans that now became the templates for the next round of synthesis. So when we denature in cycle 2, these two beyond pecans are going to be single-stranded molecules that will serve as templates. Then we anneal. And now, once again, the primers are going to bind only partially. There's still going to be these tails flapping around, not bound to DNA. Why? Because this region here is still a copy of what was present in that original DNA molecule, and this restriction enzyme site was not there. So normally you would have the primer bind fully to the beyond pecan in cycle 2, but not in this case. In this case, there will still be some flapping of these tails. But like in cycle 1, what matters is that those 3' hydroxyls are still there available for Taq polymerase, because then Taq polymerase can start synthesis from there. Now, here's where the magic comes in. Towards the end of cycle 2, what's going to happen is Taq polymerase didn't know that this was not part of the original DNA sequence. It didn't know that it was something that we as human beings artificially introduced into those primers. It will use it as a template, read it as a template, and write the complementary sequence as it should. So by the end of cycle 2, and as Taq polymerase went through the entire beyond pecan and fell off when it reached the end on both sides, it will have written the complementary sequence to that restriction enzyme target site that we introduced in the primers. And like when we were using PCR to introduce a point mutation in a sequence of interest, from there on, PCR will continue copying these sequences. This time it's not one wrong nucleotides. It's a whole collection of 6 of them, but it will continue copy them because obviously PCR doesn't know that we introduced that there. So from cycle 3 and onwards, there's going to be now full annealing of the primer with the RE side included. And from there on, with each cycle of extension, there's going to be a copying of the RE side into the amplicon. So starting in cycle 3 and onwards, we're going to get this is the amplicon that we will obtain. Once again, yellow is the sequence that was there in the original DNA sample, and it's the sequence that we wanted to clone into our vector. That blue stuff on both ends, we introduced artificially by having those sequences being part of those primers, and they were part of the primers, but starting cycle 3, Taq polymerase doesn't even know that they were not in the original DNA, thinks they were, and it will keep copying and amplifying it, and from 1 amplicon you'll get 2, from 2 you get 4, and so forth. Yeah? So after 30, 40 cycles, we'll have billions of copies of this thing with the RE sites on both ends. So what we would do then is pretend that this thing was in an original DNA, go in with our restriction enzymes of choice, and those are going to cut, will be an insert of interest that is flanked by these protruding or sticky ends on both sides. And if we cut the vector that we'd like to introduce this into with the same restriction enzyme then we did the insert. Now we're going to get this base pairing of the insert to the vector at low temperature. Then we introduce ligase. We seal those missing phosphodiester bonds, and forever will this insert be part of the vector. Yeah? So let's say, we'll go back to our example. Let's say that, as usual, there are no RE sites flanking our insert of interest, but we can introduce them through this modified PCR. So now we design primers that will flank or bind our insert of interest and introduce those Hindi3 sites. We amplify the primer by PCR, adding the Hindi3 sites. Then we do all of this cutting and pasting that I mentioned before, right? What we do is we cut the vector open using Hindi3, so now the vector will be open and we'll have sticky ends for Hindi3 on both ends of that open cloning vector. The insert of interest will have been amplified by PCR, including Hindi3 sites on both ends. We cut them, so generating those sticky ends, and then we mix everything in a test tube at low temperatures. We let these base pairing bring the insert and the vector together, and then we add some ligase. And then when everything has been ligated, we can expose it to bacteria and hope that some of the bacteria will pick it up, and we grow them on a plate with an antibiotic, let's say ampicillin, and if the vector had ampicillin resistance, then they will form colonies. Everything else will die because they cannot resist the ampicillin, but those that picked up the vector with the ampicillin resistant gene and our insert will have grown, right? Now, sounds easy. And yet, when we're doing this for real, we still don't know if these colonies actually picked up the quote-unquote right plasmid. And what I mean by right plasmid is two things. In an ideal world, everything works as I just mentioned, right? You have your open vector, you have your insert, the insert goes into the vector as you expected, right? Now given orientation, from, let's say, with the coding sequence for leptin, if you remember the example from last week, from left to right, so everything is great. That's in an ideal world. In a real world, there are two problems associated with this. Problem number one is that when you pay attention to the sticky ends of the open vector, you'll notice that they're complementary to each other too. We made it look like, or we made it sound before, like the sticky ends of the vector will find the sticky ends of the insert and they will base pair with each other, and the only way that you could have this base pairing is between vector and insert. But when you look a little closer at the sticky ends in the open vector, you'll notice that there is still complementarity, T to A, C to G, G to C, and A, even though it got a little cut out, to T. So when we mix everything in a tube and we lower the temperature, there will be many vector molecules that instead of base pairing with the sticky ends of the insert will base pair with themselves. And that will lead to something we call vector recircularization, meaning that the vector will open up when we digest it with the restriction enzyme, but then close back up when we ligate it. Right? No insert in it. It's an empty vector. We've recreated the empty vector. Problem number two, and you can appreciate it when you look a little closer at the sequence of both protruding ends in the insert. If we cut with the same enzyme, the protruding ends on both ends of this insert are symmetrical. So if we invert the insert sequence, we'll see that once again we've recreated that complementarity between the protruding ends. So A to T, G to C, C to G, T to A, and same here, T to A, C to G, G to C, and A to T. What that means is that if we mix the insert an open vector in a test tube that is chilled, the base pairing that we want can happen in the orientation that we wanted, but it can also happen in the reverse orientation, right? And we haven't really discussed this in much detail, but often what happens is you need these inserts to go into your vectors in a given orientation, because we will use these vectors to use the sequences that we inserted into them in manners in which the orientation of the DNA matters. For instance, if we want to express this gene in this insert in cells, we need the gene to go in the right direction. If not, the transcription machinery will read the gene backwards and will generate an RNA that is not going to have the sequence of letters that we need. Okay, we'll hang on to that thought. We'll revisit that concept next week. But for now, let's just go with the idea that inserts often should go in a given orientation into your vectors, and when you have these restriction digests with only one enzyme that generates the same sticky ends on both ends of your insert, the insert could go into your vector either way. So those are two very common problems. So let's go through how we solve that problem. Because when you mix your ligation reaction that was in a tube with the bacteria, there will be some bacteria that pick up empty vector, some bacteria that pick up a vector with the insert in the wrong orientation, and then some bacteria that will pick up the insert in the right orientation. And those are the ones that you want. So how do we distinguish among those? We can take two approaches, and I'm going to have you think about some of these things. So here's the exercise. We have our plate with the colonies in it. So we know that all of those bacteria picked up a vector because they're surviving in a plate with ampicillin or some other antibiotic, meaning that they picked up the vector. Now, is it the right vector? That's what we need to determine with some molecular biology afterwards. So the idea here is we will let the bacteria grow and make many copies of the vector that they picked up, right or wrong. Then we'll isolate those plasmids from never mind the Y colonies, I reordered the slides, that will make sense a little later. So we'll isolate plasmid DNA from colonies, and then we'll digest those vectors that we isolated from the bacteria with enzymes, restriction enzymes. And what I'm doing here is showing you the maps of where those restriction enzymes would cut, depending on whether a vector is empty, and that would be the size of the empty vector. This empty vector is 4.7 kilobases, or 4,700 nucleotides in length, where they would cut if the vector went in the right orientation. So there would be one cut at the beginning of the insert and one cut at the end of the insert. And then what would happen if the vector went in the reverse orientation? So again, one cut just before the MCS, multiple colony site, and then because the insert went in the wrong orientation, the second target site for the enzyme is a lot closer to the first. So what I'm asking you now to do is to think what would happen if we extract the plasmid DNA from each of those colonies, we digest them with enzymes that will generate different fragments, and run an agarose gel, run one of those electrophoresis that we discussed on Tuesday. So think about it for a few seconds, then consult with neighbors, and what do you expect to see? Now there's been some conversation. Let's do it together. So let's start with the easy one. Let's say that we picked up a colony that was surviving on that plate because it had been transformed by empty vector. We isolate that plasmid from those colonies. And by the way, that colony has millions of bacteria in it. So when we isolate the plasmid from it, we will have millions of copies of this plasmid. I mentioned that because it's important to understand that when you run those things in an agarose gel, like when you were running PCR amplicons, you will be able to see them because there's millions of copies of this vector insert, and so forth. So let's imagine that we pick up a colony that, unbeknownst to us, had picked up an empty vector. It was surviving on the ampicillin plate because it had empty vector, but no insert. You treat it with the RE that cuts only in that place here. So my question to you is, after your digestion, how many DNA molecules will you have? You started with a circular DNA molecule, and you cut it with the restriction enzyme in one place. So how many DNA molecules will you have? One. The only difference between before and after the digestion is that before the digestion, that molecule was circular. After the digestion, it will be linear, right? It'll be open, but it will still be one DNA molecule. And what size will it be? 4.7 kilobases. With these restriction enzymes, we introduce these cuts, but as I think I was mentioning before, there's no chewing up of any additional nucleotides. It's just a clean cut, right? So we're opening up this vector, making it into a linear molecule that is 4.7 kilobases. So if the plasmid DNA from that first colony we had loaded in this first well here, and we run that digestion reaction, or the product of that digestion reaction in the agarose gel, where do you think the band will be? Between 4.5 and 5, right? So somewhere here. And it will be only one band because you have millions of vector molecules that have been cut open, and they will all be the same size, and they will all run together to the same spot. Now that we did it for the simplest of examples, let's do it for a slightly more difficult one. So let's say now that you pick up a colony that, like us, had the right vector with the insert in the right orientation. You digest that vector with insert with the same restriction enzyme, except that now the restriction enzyme has a target site in the vector itself, like it did in the previous case, but it turns out that your insert of interest also has a target site for it. So now this vector plus insert has two sites for this restriction enzyme. So after the digestion, how many fragments of DNA will you have? Two. And what will be their sizes? Now I hear again 3.2 and 1.5, you said? So 3.2, where does the 3.2 come from? The insert itself, right? And where does the 1.5? Oh, sorry. 3.2, not 3.5, 3.2. And the 1.5 is the vector? Is it 1.5 only? It's 4.7. It's 4.7. So what was 3.2 is the insert itself. But then the vector is still 4.7. Do you see that? When we cut the vector open, we haven't really chewed away any of its sequence. We've just cut it open. And then we inserted something that was 3.2 into it. The size of the whole thing is not listed here, but the size of the whole thing will be 4.7 plus 3.2 for a total of 7.9. That's the whole thing, vector plus insert. But because with this restriction digest, we're cutting the insert out, we're basically reconstituting an empty vector and the insert by itself. So 4.7 and 3.2. And what would that look like in a gel? What would it look like?
One would be at the same place.
Same place.
And then one would be a little bit below the 3.5.
That's right, something like this, right? Cool. And what would happen in the last scenario? Let's say that we pick up a colony, unbeknownst to us, unlucky us, that colony had picked up a vector with the insert in the wrong orientation. We isolate that plasmid, we treat it with the restriction enzyme, and it turns out now that the restriction enzyme will find two target sites in this vector with the insert in the reverse orientation. One in the what we call the backbone of this vector, the original sequence of the vector, and then the other one towards the end of the insert, but because the insert is in the reverse orientation, now they're really close to one another. So what do you think will happen then?
Would vector be the same still?
The vector now will be the 4.7 plus the insert. We're assuming here that they're kind of next to one another, yeah. So now, if the insert went in the wrong orientation, first, we will have a very long molecule that corresponds to the entirety of the vector plus the insert, and then whatever bit there is between those two are E sides. So for argument's sake, let's say that is the 7.9 that we said before, but let's say that is 7.8 and 0.1 or something like that. Right? So what would you see in the gel? You would see a band up here, and then if you're lucky, you will see a band down here, but chances are that that band is so small that will probably run off the gel before you can catch it. Did that make sense? By the way, I'm going to upload these slides to Canvas later. I have a version here that kind of reproduces what we were saying, except that this guy here should be much lower. So never mind that band there. And this one, based on the example that we were mentioning before, should be a little higher. So go by the previous slide. Okay? So restriction digests can be used not only to clone an insert of interest into a vector, but then also to run some diagnostics, right? To see if the insert of interest went into your vector in the right orientation, we can run what are called diagnostic digests. Okay? So if you ever encounter that term, diagnostic digests, that's what it means. It's taking a vector that you hope has an insert in the right orientation from bacteria, and then using these tricks of using different combinations of restriction enzymes to see if you see a banding pattern that makes sense with what you expect. Okay. There's another approach. That was the diagnostic digest is one approach. Another approach is by PCR. Right? So once again, I'm going to ask you to think a little bit about how we could do this. The scenario now is the same. We have all of those colonies on a plate. We know that some of those colonies may have picked up empty vector. Some of them may have picked up the insert in the right orientation. Some of them, the insert in the wrong orientation. What we'll do is we'll isolate those plasmids from DNA, isolate the plasmid DNA from the colonies, never mind the white, and then run PCRs with very special primers, and then run the gel. My question to you is, how would you design your primers? Where would you place your forward and reverse primers to determine if A, the insert is present in your vector or not, and B, in the right orientation? So again, I'll give you a few seconds to think about this. You need to design forward and reverse primers. Tell me where you would place them. So where would you place those primers? How would you design them? Next to the restriction enzymes. Okay, let's zoom in, and I'm going to ask you to walk me through your idea. So let's say that the original restriction enzymes were here and here, right? So when you say next to them, would you place them on the outside or the inside? Outside. On the outside, so one here and one here? Yeah. Yeah? Okay. That's one idea. We design the forward and the reverse, flanking the insert on the outside. So, do you care which one is which? No? So let's say the forward outside. So the forward outside and the reverse inside. Yeah? Okay. So that's idea number 2. Any other ideas?
Would they both have to be inside?
Both inside? We'll figure out.
Like primers and
In a way, that's what we were saying on Tuesday, right? That if you want to use PCR to determine if a sequence of interest is present or not in a place, you would design primers flanking that sequence and then ask the question, do I get any amplicons, right? So in that sense, you could design the primers on the inside of the insert of interest. That is actually the most intuitive answer to this problem. So if we do that, and I have a slide for that, it's great because if the insert is there, we'll be able to amplify by PCR. The problem, however, is that it won't allow us to distinguish if the insert went in the right orientation or the inverse. Because either way, the forward and the reverse will be binding to both ends of the insert, irrespective of in which orientation it is, and it will produce a PCR amplification product. So if we do that, if we do the forward and reverse inside of the insert, we will get bands that correspond to the insert size, by the way, on both colonies that picked up the insert in the right orientation and those that picked it up in the reverse orientation. On the other hand, if we were to do the trick of one outside and one inside, then what happens is if the insert went in the right orientation, you will have your forward and reverse facing each other, right? Because you've fixed the position of the forward, and then the reverse will be facing the forward only if the insert went in the right orientation. If the insert went in the wrong orientation, now what used to be a reverse looking back is looking in the same direction as the forward. And when both primers look in the same direction, PCR won't work. PCR works only when the primers face one another. Your idea of having them both outside would work, except that it once again won't let us distinguish between right insertion or reverse insertion, because I didn't have a slide for that. But let's say that you have your forward on the outside, your reverse on the outside. If there is no insert, then the primers will be really close to one another, and you would get PCR amplification of a tiny, tiny amplicon that would run really fast in your gel, all the way down here, probably get off the gel. Right? If there is insert in the right orientation, you will have a much longer amplicon that goes between the two. But if you had it in the wrong orientation, you would still have, let's see if I can do that. Yeah, you will still have amplification between the two, and you would get an amplicon of the same size, right? And it wouldn't allow you to distinguish between the two. Did that make sense? So whether you have them both inside or both outside, you get amplification, you will get an amplicon if there is an insert, but it won't allow you to distinguish between the two orientations.
Would it make sense if we did like part of the vector on one side and the insert so that it wouldn't be able to read it?
Awesome. So I think you're saying this. Let me see if I understand correctly. So I'm going to redraw here. But what you're saying is you have your insert. I'm doing it in green, right? So your insert is here and the rest is vector. What you're saying is design a forward primer that sits half and half. Beautiful. Yes, absolutely. Because if you did that, when the insert went in the wrong orientation, the meaningful half of your primers wouldn't be able to base pair. I like it, yeah. That would work too. Yes. Okay? So these are, there's two reasons why we went through some of this stuff. One is because this is something that you do routinely in molecular biology labs where you do a lot of cloning. You're constantly checking the insertion of your DNA of interest into a vector, right? And I understand that not all of you will do this in your careers, but it's something that we normally do, so something that we learn. And secondly, because you may encounter some problems in quizzes and exams that have to do with this sort of puzzle approach to cloning. What happens when you have the insert in this orientation, that orientation, the primers are here or there, and so forth. you'll get to practice some of that in problems. In real life, though, one other thing we can do, and I'm going to try and get through these slides quickly, is try and avoid those problems to begin with. So for many years now, there have been more modern versions of these cloning vectors that use this very clever trick. They introduce, and you had one of the quiz questions actually related to this on Tuesday. What these modern vectors do is they have the MCS, the multiple cloning site, embedded right in the middle of the coding sequence for an enzyme. This enzyme is LAC-Z. If it rings a bell, it's because it's the same LAC-Z gene in the LAC operon, so it's beta-galactosidase. So someone put the gene for beta-galactosidase in a vector and managed to write an MCS within the coding sequence for LAC-Z without affecting its ability to express beta-galactosidase. So the idea here is, if you don't touch that MCS, or if you open the vector and then the vector recircularizes without picking up any insert, the sequence for LAC-Z will be intact. And that plasmid will be producing, in addition to the enzyme that destroys ampicillin, will be producing a lot of beta-galactosidase. And if you offer bacteria this particular compound called X-gal is a molecule that looks like lactose, except that instead of being galactose and glucose, it's galactose and some funny chemical. It can still be cleaved by beta-galactosidase, and it will produce galactose and a blue compound. It's a mouthful, but it's 5-bromo-4-chloro-3-hydroxindole-1, okay? I'm not gonna ask you to repeat that, don't worry. It's blue stuff. So the bottom line here is that if you use one of these vectors and your insert went into the MCS, it will interrupt the normal coding sequence of LAC-Z. No beta-galactosidase will be produced by those bacteria, and then this funny chemical cannot be turned into the blue product. So those bacteria that picked up a plasmid with an insert inside of the MCS will not produce beta-galactosidase, will not produce blue product, will make creamy colonies or white colonies, and hence all of those references in the previous slides to the white colonies, right? On the other hand, if a bacterium picked up a recircularized empty vector, in those bacteria, LAC-Z will be intact, beta-galactosidase will be produced, that funny X-gal compound will be degraded, they will make this blue compound, and they will turn blue in your plate. So when we use these more modern vectors, and these are being used every day in labs nowadays, you can basically plate your bacteria in the presence of ampicillin for selection and X-gal for selection of empty vector or vector with insert. The day after, you see all of these colonies on the plate. Some of them are going to be creamy white, some of them are going to be blue. And you simply avoid the blue ones because those picked up empty vector. And for the second problem, the problem of inverse orientation of the insert, we go back to, I think there was a question before, if we can use a combination of restriction enzymes, and that's exactly what we do. That is called directional cloning, and the idea here is, instead of using only one restriction enzyme to cut out your insert from its source and cut the vector open, you use two. And you use two because, generally speaking, you will be able to very quickly find a combination of enzymes such that they cut asymmetrically, but in a way that is not in the same orientation, in the same manner, such that when you try and insert the DNA of interest into the vector in the right orientation, it will go in, right? So this thing base pairs just fine. But if you turn the insert around and you try and do that base pairing, if we zoom in and take a closer look, we'll see that there is no correspondence of the nucleotides anymore, right? So there's no way that there can be base pairing between the insert and the open vector if the insert flipped upside down in space. So simply by digesting vector and insert with two different enzymes, chances are that the only way that the insert can go into your vector is the orientation that you want.